Okay, we're going to be starting Unit 1 PowerPoint 2 Part A. And a large portion of this particular PowerPoint is all review and material that I would expect you to know from previous biology courses. I did post a Bio 181 review PowerPoint within the module at the bottom that you can review, which talked about the different macromolecules and enzymes and other metabolic things. So today we're doing essentially page 2 and the beginning of page 3 on the lecture outline if you want to follow around or follow along. Everything here should be something that you are at least vaguely familiar with. So right here, this structure is glucose. Glucose, when we talk about things like metabolism, is traditionally the food source. Here, this is DNA, the double helix and double-stranded helix. Here we are looking at a protein that is in either the tertiary or quaternary form, which is level 3 or 4. Here, these are also proteins. This is a disaccharide. It's specifically lactose, so if you compare the two, this is a monosaccharide, 1 sugar versus 2 sugars. Here we have a phospholipid. These are the building blocks of the plasma membrane or cell membrane, not the cell wall, the cell membrane. And here we have a phosphate group, a base, and a sugar. So you should know how that picture relates to this double-stranded DNA helix. And finally here on the bottom, we have 3 phosphate groups, and then we have a sugar, an adenine, which is a base. So this is actually ATP, which is adenosine triphosphate. That is your energy yielding molecule, which then breaks down to ADP, and pulling off that 1 phosphate group releases energy for chemical reactions to occur. Please review the various macromolecules. I traditionally give a homework assignment for this, but I think the PowerPoint for 181 is enough to help everybody. The 4 macromolecule groups are, there's proteins, carbs or carbohydrates, nucleic acids, that looks bad, sorry, and fats. So these are the 4 groups. You should know what their functions are, be able to attach a picture to it. When you take your exam, there'll be several pictures shown, and the 2 that I particularly focus on are nucleic acids and proteins. So when you take it a picture, you're going to have to know if it is a protein or a nucleic acid. The next aspect when it comes to growth of microbes has to do with metabolism. When you take a look at this picture, you should probably or will probably be having some type of nightmares about regular biology classes, whether it was the 156 or the 181, where you went through all of the breakdown of ATP to ADP and NADPH to NADP+. Lucky for you, I do not expect you to go back and remember this entire chart, but what I do want you to be familiar with is both anabolism and catabolism. So metabolism consists of every single metabolic pathway. Without enzymes helping to do this work, organ systems would simply shut down in both humans and every other type of microbe. An easy way to remember this, anabolism is taking smaller components and building large macromolecules from them. And if for AN you remember or think of anabolic steroids, which then make the muscles grow, so anabolism is the making of larger macromolecules, and catabolism is taking those large molecules and actually making them smaller and breaking them down. Together with anabolism and catabolism, you have metabolism as a whole. So again, this slide is nothing more than review. Everyone should recognize that these 2 pictures are, again, proteins. I would be asking you in class what this guy is over here, but essentially it is glucose. This is the phospholipid, which is a building block of the plasma membrane or the cell membrane. Remember that this plasma membrane is known as selectively permeable which means there are charges and size differentiations. Not everything can easily flow within and out of this plasma membrane because it is known as selectively permeable. Something like water can in fact go in and out. This right here is DNA, your double helix, and here you can see it broken up with the bases where cytosine pairs with guanine and adenine and thymine. This is actually a bacteria, which is going to be the focus of our class. So with everything, you should just be recognizing them, and very importantly are enzymes. This is an important part of our class, so please make sure you review them. If you don't remember things like this being the active site, this being the substrate, competitive and non-competitive inhibitors, how it has a locker key function that when the substrate enters the active site, it squeezes and locks itself down to hold that in place. So we're going to talk a little bit about enzymes. So here you have an actual enzyme. The opening area is known as the active site, and what's going into that area is known as the substrate. If you increase the amount of substrate, you have a higher binding between the enzyme, the active site, and the substrate. So enzymes, essentially, simply put, make the world go round. Without enzymes, all of metabolism would shut down. They catalyze or speed up all chemical reactions, and this occurs without using up the energy. So let's take a look at the picture on the bottom here to see if you remember what's happening, because there are in fact two types of inhibition that you need to be familiar with, and this is how enzymes are regulated or controlled. So again, you have the enzyme, and this right here is the active site, and you have a substrate that is binding. So in this case, this is your primary substrate that you want to bind, but instead this little square got in the way. So this is known as competitive inhibition because it competed and locked out this particular substrate from binding, allowing that particular enzyme to do its job and catalyze a reaction. Whereas if you increase the substrate concentration, and again, these are the primary substrates that you want to bind to the active site, that does in fact happen, and in this case it's considered non-competitive because it didn't get into the way. And therefore, you have competitive and non-competitive, and this is how enzymes are regulated. If you take a look at the picture on the top, this is showing a primary active site, and this is potentially a secondary active site. So one does not necessarily affect the other, except for when this substrate binds and the entire enzyme does the lock and key squeezing, it can change the shape or conformation of the primary binding site, stopping that enzyme from attaching at the substrate at its active site and doing the job that it needs to do. So how are these controlled? Well, enzymatic pathways and metabolism are essentially controlled by feedback inhibition. So if you take a look at this picture, again, you have enzymes on both sides, and then you have the substrate binding. When the substrate binds, and it binds properly and there's nothing in the way competing with it, it allows this enzyme to work through its pathway and keep operating to catalyze enzymatic reactions. Whereas when a secondary or different substrate binds and acts in a competitive manner, then it's going to shut down the pathway and inhibit it from actually working. So in simple terms, to explain this, if you read what is written on the bottom, a method of metabolic control, or to control metabolism, which is again anabolism and catabolism, what you look at is the end product of the pathway acts as an inhibitor, and when something acts as an inhibitor, it shuts down a pathway. Oftentimes microbes do this to spare energy. It needs to make a certain amount of end products for nutritional or other needs, but when enough is made, the competitive inhibition, also known as allosteric inhibitors, comes into play to shut that particular pathway down so that extra energy is not wasted. So we've been talking about ways that enzymes control and regulate things in microbial growth. there are other conditions that need to be met for microbes to grow, and one of them have to do with temperature ranges. So with temperature range, or with the growth of microbes, there is always an optimal temperature. An example for this is, there is an organism called Bacillus subtilis. It tends to grow in the soil. So if you were to grow this up in the lab, you would put it in an incubator that's similar to room temperature, because that is the optimal growth for that bacteria to propagate and grow on the plate for you to work with in the lab. Let's say we isolated something like E. coli, which we know grows in the humans. Human body temperature is 37 degrees Celsius, which is the same as 98.6 degrees Fahrenheit. In science classes, we never talk about Fahrenheit. We really do stick to Celsius. And therefore, if you wanted to grow Staph aureus or E. coli that we know grow in and on the human body, we would want to use an incubator that is around 37 degrees Celsius, because again, growth of microbes always has an optimal temperature range. So you need to be familiar with 4 of the terms that relate to these temperature ranges for the growth of microbials. If you look at this chart, the x-axis is temperature in Celsius, and the y-axis is the rate of growth. So these different terms are psychrophiles. If you look at the temperature range on the bottom here, minus 10 degrees Celsius is incredibly cold, so think of the Antarctic. Then we have mesophiles. Next we have thermophiles. And then there are hyperthermophiles, which are somewhere between 68 degrees Celsius and 110. So these are found, for example, at the bottom of the ocean hydrothermal vents. These are incredibly high temperatures. Expect to see this material when it comes to exam time using a little bit of critical thinking. If I tell you that an organism was isolated that was a psychrophile, would that grow in the human body? And the answer is absolutely not, because the range for psychrophiles are from minus 10 to approximately 18 or 19 degrees Celsius, whereas our body temperature is somewhere right here, which is about 37. So essentially, bacteria that grows in us are in fact going to be mesophiles. Is it possible that a thermophile can grow or be isolated from the human body? It is possible, because if you look, a thermophile is around 40 degrees, and we've already said that we, as a human, are at 37 Celsius, so that's close enough. I want you to ponder the question of whether or not we can isolate a hyperthermophile from a human, and hopefully you know the answer is no. So this picture is just a little bit cleaner without all of my scribbling. Okay, so on this picture here, I would like for you to apply the correct terms. If a bacteria or microbe was isolated from the human body, it would in fact be at around this optimal temperature. And the term for that would be mesophile, because this is our range. The psychrophile and psychrophile down here, that's very cold, and hyperthermophile. This picture are those hydrothermal vents at the bottom of the ocean that I mentioned. This right here is the grouping for thermophile. So is it possible that this actually be found in a human? Not likely, but it is in fact possible. So again, these are optimal temperatures. So now we have to relate temperatures to enzymes. Hopefully, again, you're remembering that this is where human body temperature is. So here are our mesophiles with enzymes, and I might be aging myself here, but there used to be a commercial back in the 80s, and they took a frying pan and they put an egg into this frying pan and fried it, and then the commercial, it was a public service announcement, basically said, this is your brain on drugs. So if you take an egg that is actually coming out of the fridge, you crack it and you throw it onto a frying pan with hot oil in it, you have now essentially denatured that protein. There is no undoing that. You cannot take that egg out of the frying pan and put it back into the shell. On the other hand, if you take a cold egg out of your refrigerator and put it on the counter, after a little while it starts to perspire and there's the water on the outside. You then technically could take that egg and put it back into the fridge because you didn't fry it in a frying pan. So putting it back to its regular shape is called renatured or renaturation. So what you're looking at here is you have an enzyme, and one way to denature an enzyme is based on temperature or heat. Why is this important? Because as we mentioned previously, enzymes completely rule and run the concept of metabolic activity in all living things and all microbes, all humans. Without metabolic activity, all the processes shut down. Another aspect to temperature has to do with food spoilage. So if you take a look at this chart, the bottom is in hours, so time frame, and the y-axis is in temperature. I have a very small household, and I tend to shop a lot at Costco. So if I were to buy a tray of pork chops or steaks, I can't take the entire thing and freeze it, and then de-thaw the entire thing, cook a couple, and refreeze it, and keep going back and forth, because I'm going to harbor the growth of microbes. So what you would do is you would separate that food and de-thaw it. Once you freeze and unfreeze, you cannot refreeze food. And this all has to do with optimal temperatures as it comes to bacterial growth. So another thing to consider, if you have a large vat or pot of soup or chili or something, you can't take it when it's warm and put it into the refrigerator. You need to let it cool to room temperature first, because otherwise that heat and then putting it in the refrigerator, you are harboring bacterial growth. So if you take a look at this picture here, this yellow area here is the temperature range or optimal temperature for an organism called Bacillus cereus. This happens to grow very easily in rice. So this little black line on the bottom here is the refrigerator air, which tends to be between 0 and 6 degrees Celsius. Again, this yellow area here happens to be where that bacteria harbors growth really well in rice. And then you have two different dishes, the blue one, which is a little bit smaller, and then the pink one, which is a little bit larger. If you were to put the smaller bowl into the refrigerator, because the whole concept of the refrigerator is that it will allow food to not get as spoiled, notice the blue line is dipping quite a bit because it's a small container and the food is able to slow down or cool down a lot easier, and then very quickly it matches refrigerator air, and therefore the growth line for this blue one here is below where Bacillus cereus would replicate, and therefore the food will not spoil. But if you take a deeper dish, notice that the plunge of the line is a lot slower. So it stays within that particular area that is the optimal temperature for the growth of Bacillus cereus, and therefore the food has the ability to harbor that growth even within the refrigerator by putting it in a warm setting. Next, it's not just temperature, because again, enzymes are regulated by many things. Another example is pH levels. There is, in fact, an optimal pH level. The pH scale goes pretty much from 0 to 14. 7 is considered a neutral temperature. Our GI tract or our stomach area is at a lower temperature. The closer you get to 0, below 7 is considered acidic. And moving from 7 and above is considered basic. So the categories of bacteria are either acidophiles, which have a pH less than 7, neutrophiles, which are at about 7, because again, that is neutral, and alkalophiles, or acidophiles is another name for it, and these have a pH that is greater than 7. Why is this important? Because when we look at growth of organisms, not everything grows everywhere. So take a look at this chart on the bottom. If I asked you if the organism Enterococcus faecalis is likely to grow in your GI tract, what you would do is take a look and you see that that is coded by the color red. The color red is a pH 7. Our GI tract is closer to a pH 3. So no, it would not grow there. If you take a look at something like Saccharomyces cerevisiae, which is coded by green, that is actually yeast. Notice that it grows more in a below 7 pH level, meaning it is an acidophile, or a lover of acids and has the optimal desire to grow in an acidic environment, and then pretty much stops at a pH of 7. And then you can also look at the bacillus, which is in yellow, and the bacillus tends to grow in both acidic, neutral, as well as an alkalophile or a basic type of environment. So to put this together with both temperature, if I were to ask you to give two terms related to an organism that was isolated from a human's GI tract, two terms that you can already relate to that particular organism are that it is a mesophile, and you know that because it came out of a human, which is in that 37 degrees Celsius range. And the second thing you can attach to it is you now know it is also an acidophile. Why? Because the GI tract is around a pH of 7. So, two optimal things, temperature as well as pH. A couple quick examples of that are the vaginal area is acidic at a pH of 5, and you know it's acidic because it's less than 7. So lactobacillus growing on a vaginal cell is an acidophile, and Vibrio prefers an alkalophile or alkaline-based area because it is above pH of 7, and therefore its pH requirements are more basic than acidic. Next, we're going to mention membranes a little bit. These are selectively permeable, as I had already mentioned previously. So what you should see here, all of these little red and white things, these are all essentially water. So notice that water is located both inside as well as outside of this membrane. On the bottom, you see salts, proteins, and sugars. These are larger macromolecules, and because this membrane is selectively permeable, these are not able to get through the membrane without some type of input of energy or facilitated diffusion. Water, on the other hand, is through osmosis. It's constantly going to go in and out. Another thing to consider is to look at this drawing, which is trying to depict all of the phospholipid bilayers or the phospholipids that actually make up the bilayer of this selectively permeable plasma membrane. So at this point, we need to look a little bit at osmotic pressure and moisture. So I'm just going to give you the terms and ask you to take a quick peek at an older PowerPoint review that I had posted, or Google if you prefer. But what I need you to remember are hypertonic and hypotonic as well as isotonic. So what's essentially happening here, if something is hypertonic, the solute levels are incredibly high within the solvent. Solvent is traditionally water. And in hypotonic, the solute levels are very low and the water is very high. So hypertonic would almost be like a raisin where the water levels are less because there is more solute. And hypotonic, the water's more, so this is almost like making Kool-Aid or Crystal Light and not adding enough solute or powder to the solvent, which is the water. This comes up because it's important that you realize that bacterial cells prefer a slightly hypertonic condition, which means a little bit wrinkled, raisiny, not completely blown up. If you think about people who do those balloon animals, if you blow it too hard and then you start twisting it and having it move around, they tend to pop. So they leave a little bit less air in them. Strange way to relate it, but it tends to help students. So please remember that bacterial cells prefer slightly hypertonic conditions, which means more solute in a little bit of less solvent. This now relates back to bacteria because both bacteria and fungi have the ability to tolerate a large range of solutes. In other words, their tolerance levels are pretty good. Something like sugar in jelly and jam, once you open jam or jelly, you don't need to put it in the refrigerator because it has such a high percentage of sugar that will actually keep it from going bad. Just like when you dehydrate meat and make jerky, it's a high salinity or salt content. But it is important to remember that salt and sugar can be used to control the growth of bacteria. Too much or too little can either enhance or inhibit the growth. How does this relate all the way back to proteins? Here is your original protein. If you gently denature it, which again you can do with temperature, you can do with pH, and now I'm telling you you can do with things like salt. If it's gentle, it renatures back into its original form. If it's not gentle and too extreme, then that is the egg in the frying pan where you have denatured it to the point where it will not go back to its original shape. So halophiles are a group of bacteria that live in very extreme environments. They don't cause disease because the humans are not in that extreme environment. And if these are hyperthermophiles and they are not able to grow within us if we're talking about temperature. But for halophiles, it actually has to do with salt. These are salt-loving bacteria, whereas many bacteria adding additional salt will actually kill them because it's outside of their optimal temperatures. The last thing I want to mention and related to growth is oxygen levels. So we've already talked about temperature optimal conditions, we've talked about pH, we've talked about salts, and now we're going to talk about oxygen. So the term obligate, which you can see here in A as well as C, if something is obligatory, that means it is mandatory. I'm mentioning these two first because they are the simplest to understand. If it is an obligate anaerobe, that means it requires oxygen to be present for growth. If it's an obligate anaerobe, that means that it will die in the presence of oxygen and prefers to grow without any oxygen present, or can only grow. We're not going to look at the last two. We're going to focus on the first three. Most of the organisms that we cover in our class fall under the category of facultative anaerobes. This name might be a little bit confusing. So these bacteria that are facultative anaerobes can grow with and without the presence of oxygen, but the key is they prefer to grow with oxygen present. It's explained here on the bottom. So you have growth throughout the entire tube, whereas on the first one, if it is requiring oxygen, notice that the growth is towards the top of the tube, closest to where oxygen exchange is happening. If it's an obligate anaerobe, the growth is towards the bottom, as far away from oxygen as possible. But please note that with a facultative anaerobe, there is growth throughout the entire tube because they can grow in both conditions, with and without oxygen, but prefer oxygen to be present. So the last point I'd like to make to tie together all of these requirements are if you have an organism that is isolated from the GI tract of a patient, name 3 terms that can relate to it. Well, we know the GI tract is acidic, so you know that's an acidophile. We know that the patient in humans has 37 degrees Celsius as their body temperature, which tells us that they are mesophiles. We know that certain oxygen level requirements, and I told you 90+% of the bacteria we're going to talk about. So you also can say they're facultative anaerobes, which means that they can grow with or without oxygen, but they prefer the presence of oxygen. So how would you have figured this out? Well, the GI tract gives you the information of the pH level. The fact that it's a human patient should give you the temperature level, and being that it's in the human body would tell you that this is a facultative anaerobe because we aren't fully aerobic or anaerobic. So please look over these review things. If you have a hard time with enzymes and metabolism and pH and hypo tonicities, remember bacteria tend to be slightly hypertonic, which essentially means that the solute level is higher, which automatically will make the solvent level a bit lower, meaning they're a little more raisin-like, not overblown up. Please review these things. It will help you be successful in this first unit. Most of this has been covered in other courses, minus the terminology related to specifically microbes.